In AACR, actually, we have brought new data about an animal model which make use of a very known gene that is named RET, that is implicated in 5 different human diseases, including medullary thyroid carcinoma. And what we have shown is that there is a genetic control of the penetrance of this gene, in the sense that in a particular strain of mice, we have 100% of tumor, and in another one we have 0. So we are trying to identify the genetic elements who controls the penetrance of the gene. That was basically what we have presented. And of course, once these genes have been identified, they can be used also in human. We are trying to develop a quite novel approach to the so-called personalized or target therapy. That means that we have elaborated a 3D model of the kinase domain of a couple of receptors, in particular c-KIT and PDGF receptor alpha and beta. And based on the model, we can simulate in silico the interaction between a drug and this particular kinase domain, which could be altered by mutation, causing resistance to the drug. So we are predicting whether we should increase the dosage of the drug or we have to look at a new drug. And once we have the new drug, we can simulate whether it will work or not. Then we pass in vitro, moving the gene with the mutation in a cell line, treated the cell line with the same compound, and see whether it acts in switching off the kinases. And finally, we go to the patients, and it happens, now it's still anecdotal because we have to conduct more large studies, but we can see in 2 weeks some metabolic change in the tumor that apparently stop growth after providing this drug that has been identified in a way that I described before. Actually, we do believe that with this kind of approach, we have 2 expectations. One is a more appropriate use of this drug that costs a lot, but once they are appropriately provided to the patient, has a very high percentage of success, despite of the fact that most of the time we have to treat chronically the patient. And the second point is that this in silico approach can also be used in an efficient way to select new molecules before to pass to the subsequent phases using cell that are more costly.